So I just discussed with you how you would genotype a single spore with respect to a locus or alleles that you're interested in. But what's important with tetrad analysis is we're trying to understand the relationship between two genes. Okay, so the way you would do it is you would look at two genotypes. Right. So for example, say we were crossing a Lu minus line with a Met plus line. So you do haploid mating with a Met plus Lu minus. So you have these two haploid strains, you cross them together, you make a diploid, you make them do meiosis and by starvation, and then you would end up with a tetrad, right? And you want to look at the relationships, basically the linkage relationships in a way, between these two markers. Right? So the principle of that then was you would take out these 4 of these haploid spores, and you would pick them apart, and you literally would do something called a tetrad dissection under a microscope. Right? And so after that dissection, you grow those on complete media, 1, 2, 3, 4, so you can actually grow them, because if you grew them on some sort of minimal media or something, you wouldn't, the things that would be defective, for example, for the production of leucine, wouldn't be able to grow. So you can grow them up, and then what you can do with your plate that has 4 spores on them, and you're actually going to do many, many, many tetrads. So you'll have a plate basically that looks like this. You'll have 1, 2, 3, 4 growing from tetrad 1. You'll have another tetrad, another tetrad, another tetrad, 1, 2, 3, 4, 1, 2, 3, 4, 1, 2, 3, 4, so on and so forth. All right, and then you're going to genotype each one of those with respect to, for example, in this particular case, on Lu minus media with respect to the leucine gene, or Met minus media, right? And so you have basically a plate that looks like this and a plate that looks like this. And you're going to just then see if each one of those 4 grows or does not grow. So any of the ones that are Lu minus wouldn't grow. So for example, if this is Lu minus and this is Lu minus, but this is Lu plus and this is Lu plus, you get a pattern that looks like this. You get growth, growth, no growth, no growth. Right? And likewise, for the same tetrad that you've dissected, you may get a pattern like this for Met, right? So in this particular case, you get growth, no growth, no growth, growth, right? Now, we're interested in the relationships between these two genes, and in this particular case now, you can actually genotype this tetrad and all 4 haploid products. This first one here, okay, didn't grow on leucine, so spore 1, we'll just do spore 2, 3, and 4, all right? Spore 1 has to be Lu minus. It's incapable of growing on Lu minus media. This is Met minus media, all right? So it's Lu minus, but it's capable of growing on Met minus media, so we would call it Met plus, all right? Let's look at the next spore. It's either capable of growing on Lu minus media or Met minus media, so it has to be Lu minus Met minus. And it's capable of growing on Lu minus media, but incapable of growing on Met minus media, so this would be Lu plus and Met minus. Right? And in the last particular case, it grew on both Lu minus media and Met minus media, so this would be Lu plus Met plus. Now there's, in any particular mating, all right, there are going to be different types of combinations. Right? One particular combination that you could have here is that all 4 spores would show this particular pattern. So for example, you have a tetrad here, you have 2 spores that are like this, all right? And they're Lu minus Met plus, Lu minus Met plus. And then you had another pair of spores that are Met plus, Lu minus, and Met plus Lu minus. All right. Now notice something here, right? These spores come in pairs. Basically, there's 2 types of genotypes. There's the Lu minus Met plus genotype and the Met plus Lu minus genotype. So there's 2 types, and what type are they? Well, they represent the parental genotypes that you set up for this crossing. So this would be, this particular arrangement would be called a parental ditype. Parental ditype. All right. Now you can also have another arrangement in which you have two types of spores as well. All right, you have Lu plus Met plus Lu plus Met plus, and then Lu minus Met minus, and then Lu minus Met minus. All right. So again, there's two types, but they're of the non-parental type, because you can see that the association between Lu minus and Met plus has become decoupled. Now Lu plus is associated with Met plus. So this would be called a non-parental type. And then there's really only other sort of one arrangement that's possible, right? Basically, you could sort of have all different combinations of those particular genotypes. So, for example, you have a tetrad like this, in which one of them is Lu minus Met plus, right? This is a parental type of genotype. You can have the other parental type of genotype, Lu minus Met plus, right? But then you could also have sort of both of them be Lu plus and Met plus. Both of them are plus. And then the other one is Lu minus Met minus. Right? And so this particular case, there's four kinds, right? There's sort of the parental kinds and the non-parental kinds. And this would be called the tetratype.